Before I introduce Debbie and Heather and Logan, I want to tell you a little bit about Miller syndrome. So Miller syndrome is a rare condition, also known as postaxial acrofacial dysostosis. So we're kind of glad that it also has the name Miller syndrome. But the clinical description actually does tell us a lot about the condition. Dysostosis, it is a bone malformation syndrome. It affects the face as well as the distal appendages. And it's postaxial, so primarily behind the axis, affecting bones like the ulna, the fibula. So there are craniofacial malformations, limb malformations. You'll see a picture of that in a moment. Conductive hearing loss and cleft palate are all part of the condition that has been called Miller syndrome. Now, in 1977, when Heather was born, there were no recurrent cases. That is, all of the 3 or 4 cases that were known were single cases in families. So imagine that you are counseling a family in which there's been no known recurrence, only a few cases known in the world. Their question is, could this happen again? What's the recurrence risk that you would give them? What's the best estimate? 0? Well, is it 0? Could it never happen again? Actually, the best answer, and I saw a couple of you saying it, was, I don't know. And in this case, that is the right answer. When there have been only a few cases reported in the world, the genetics is not known. And the best answer is we don't know how likely this is to reoccur in a family. As you'll hear from Debbie, that's not the answer that she was given. It's kind of interesting. So here is a picture of Heather. She was written up in the Journal of Pediatrics back in 1979. And you can see some of the craniofacial malformations that help to define Miller syndrome. Also, missing digits. And her forearms are substantially foreshortened. Here's an X-ray showing that the radius and ulna are relatively short and small. Also missing digits here. So here in 1981, Heather and Logan were both written up in the Journal of Pediatrics because this was the first known recurrence of Miller syndrome in a family. It had never been seen before twice in the same family. Heather and Logan have very similar phenotypic features. And this was in 1981, this was about the time that I first learned about Heather and Logan. I used to spend every Tuesday in the pediatric genetics clinic here. I was trying to learn more about clinical genetics. And that's where I first heard the discussions about Heather and Logan, what their diagnosis might be, what their features were. Here's a photograph of Heather a little bit later. Her arms were splinted to try to sort of loosen her joints up prior to one of her surgeries. Between the two of them, Heather and Logan have had more than 50 major surgeries. In fact, Logan is having a corrective eye surgery next month, right? So no stranger to the doctor's office. Here's Heather, again a little bit later, making something with Play-Doh, I think, right? Yeah. And here's a picture of Logan as an infant. And you can see, again, features very similar to those of his sister. This is Heather at her high school graduation. Shortly after that, she had corrective jaw surgery. You can see quite a difference there as a result of that surgery. So here is Bridget, another little girl with Miller syndrome. And you can see, again, very similar craniofacial features. The limbs look very similar. So another person diagnosed with Miller syndrome. Here she's shown later with Logan. You can see that she's had some eye surgery to correct the lower lids. They sort of turn down typically in Miller syndrome. Here is the other family, a family from New Zealand, in which there has now been a recurrence of this condition in the same family. So a brother and sister, both affected with Miller syndrome, shown with their unaffected older sister. And so you can start to see very, very similar phenotypic features in people with this condition. And this is one of the things that allows a clinical geneticist to make the diagnosis of Miller syndrome. If we look at the family history, here's Heather, here's Logan, shown with their unaffected parents. In addition to Miller syndrome, both Heather and Logan, since they were very young, have had chronic bronchiectasis, where there's dilation of the airway, recurrent infection, Pseudomonas, staph, many of the same kinds of bacterial infections that you see in cystic fibrosis. So that's not part of Miller syndrome, but they've also been affected with bronchiectasis all their lives. And we knew that there was a significant family history. They had a first cousin, Brandon, who had cystic fibrosis and who, in fact, died now about 3 years ago. So given that he was known to be affected with cystic fibrosis, and by the way, CF accounts for about 1/3 of cases of bronchiectasis in the United States, so it is a major cause of this form of lung disease. So you have an affected individual here. What's the probability that his mother would be a carrier? Yeah, she is an obligate carrier, given that he is affected. So what's the chance that Debbie here would be a carrier for a CFTR mutation? Yeah, I heard several people said 50%, so that's right. She shares 50% of her DNA with her sibling, who is an obligate carrier, so she would be at 50% risk of being a CFTR carrier. So about 12 years ago, we got DNA from each family member, all 4. We sent the DNA off to my colleague Gary Cutting at Johns Hopkins, who's a cystic fibrosis expert. He tested them for CF mutations, and sure enough, all of the family members here are carriers of the major cystic fibrosis mutation delta F508, which you learned about, 3-base deletion in the CFTR gene. But the paternally transmitted copy of chromosome 7 of the CFTR gene was perfectly normal. No mutations detected. That then would not be sufficient to account for chronic bronchiectasis in the children. They also had their sweat chlorides tested. They were in the normal range, so they didn't have a classic diagnosis of cystic fibrosis. So at that point, really, the cause of their lung disease was unknown, and the genetic basis of Miller syndrome remained unknown. So for 3 decades, we had to say, we really don't know genetically what causes this condition. In fact, because there have only been 2 recurrences in the world, we can't even be sure that it is genetic. Then things started to change in around 2009, because what major technology started to become affordable? Whole genome sequencing, right? We've talked about this a little bit. In 2003, when the first human genome was fully sequenced, the total cost of the Human Genome Project, about $3 billion. A few years later, Craig Venter had his genome sequenced by his company for $100 million for one genome sequence. And then in the space of just a couple of years, the cost plummeted from $100 million to about $25,000. At that point, it was feasible to go to Heather, Logan, Debbie, and their father and get their DNA and look for every single possible mutation in their 3 billion base genome. Because the techniques we've talked about, linkage analysis, genome-wide association studies, really wouldn't be able to reveal the mutation in this family. But if we could look at every single base pair, all 3 billion of them, then we would have a chance of finding which one is actually altered. So, but I think this graph is really pretty remarkable when you look at a decline in 9 years from $3 billion to under $3,000, a million-fold decline in cost in less than a decade. There is no other technology anywhere that has seen this kind of cost reduction in so little time. So this is another graph sort of showing the same thing. This was from a review in Nature published a couple of years ago, again showing the decline in cost, and with that decline in cost, an increase in the number of people sequenced. And I want to draw your attention to the upper right part of this diagram. We'll blow it up here. And in early 2010, complete genome sequencing had been done on Archbishop Desmond Tutu, on a man named Jim Lupsky, who's a colleague of mine who has Charcot-Marie tooth disease, peripheral neuropathy, and a family of 4. So you're going to hear about the family of 4. This is a more updated graph, again, showing decline in the cost of genome sequencing, along with that an increase in the number of people whose genomes have been sequenced, estimated at the end of last year to be about 30,000. It's definitely many times that this year. So thousands and thousands of people now are having their whole genomes sequenced for a variety of research and, in some cases, clinical purposes. So what do we actually do in next generation sequencing? I don't want to go into a lot of details here because, for one thing, the technology is evolving very rapidly. But just to give you an idea of what's done, the first thing we do is to chop the DNA into small fragments, typically around 100 or so base pairs in size. So we have very small fragments of DNA. In most of the approaches, we make many copies of the DNA using the PCR technique, which you've heard about. And then the fragments are all very rapidly sequenced. And there are all sorts of ways of doing this with microarrays, magnetic beads, flow cells, nanopores. We won't go into the details, but there are machines capable of doing an entire human genome sequence in now 3 or 4 days. We then have all these sequenced fragments. We have to reassemble them into a whole genome sequence. And an advantage of what we call next generation sequencing is that it's massively parallel. You can do millions of reads all at once in a single run. A disadvantage, each read is kind of inaccurate, but what we do is we sequence every one of these fragments, every one of these regions over and over and over again, and we get a consensus read that is highly accurate. So here's the idea. We have now sequenced all these fragments. We use the original reference sequence, the one published in 2003, as kind of a map. And we map these back to that original sequence. And you can see occasionally we'll have a variant here, so this is probably a sequencing error, but we would call this a C because in most of these fragments at this position, we have a C. At this position, we see roughly half and half C's and A's, so we would call that a heterozygote. Right? We have about half C's, about half A's. And typically, here we have 11-fold coverage. Typically, we would have 30 to 50-fold. And that gives us a pretty good sequence read for that particular nucleotide position. So in essence, that's what we're doing when we're doing whole genome sequencing. So we can do whole genomes for $2,500 to $3,000. We can do just the protein coding portion, all of the exons or exomes, for about $800 a piece. And a lot of people elect to do that because we can probably get most of the critical disease information from the exome, as we call it. But the trick here is to find that one, or maybe just a few, disease-causing variants in that mass of DNA sequence. So we assess the functional consequences of each variant. Does it actually change an amino acid? If so, how does it change it? We compare it with public databases of known variants, because if it's rare in the public databases, then it's more likely to be disease-causing. And we look at other patients who have the same condition. Do they have mutations also in the same gene? So those are some of the ways that we try to find that elusive signal in 3 billion base pairs of DNA. So that was done on Heather, Logan, and their parents. We got 15 micrograms of DNA from peripheral blood cells from each family member. And then we did whole genome sequencing starting in 2009. It was done by a company in California called Complete Genomics. We had about 50x average sequencing depth, so each region, each nucleotide would be sequenced on average 50 times over to guarantee accuracy. And to make a long story short, because it took months of analysis of that sequence to get to the answer, but exome and whole genome sequencing revealed two disease-causing genes. First, the gene responsible for Miller syndrome, DHODH, encodes a dehydrogenase that's involved in pyrimidine synthesis. And this, by the way, if we had just tried to guess which genes might be involved in this condition, this would not have been a good candidate. But it turns out that in every Miller syndrome patient that has been looked at so far, they have mutations in this gene. So this is the causal gene for Miller syndrome. An interesting other discovery that was made in Heather and Logan's genomes is that they also have mutations for an autosomal recessive condition called primary ciliary dyskinesia. This encodes a dynein heavy chain, and that's needed for normal ciliary movement. You know how the cilia in the lungs beat to clear the lungs of mucus and infections and so forth. If you have that movement impaired, then you're going to be susceptible to pulmonary infections, eventually to bronchiectasis. So that accounts for their lung disease. They had never been diagnosed with that condition despite many years of having been seen in pulmonary clinics. Another thing that's often seen as a result of abnormal ciliary function is situs inversus, where the organs are misplaced, sometimes totally reversed, because you learned in embryology that for left-right orientation of your organs, the cilia have to move properly in the embryo. So in Heather, in particular, some of her organs are somewhat misplaced. That's a consequence of a mutation in the DNA H5 gene. So these results nicely then explain the phenotype. And if we look now at the pedigree again, both Heather and Logan are compound heterozygotes for mutations in two autosomal recessive conditions, Miller syndrome caused by mutations in this gene, primary ciliary dyskinesia caused by mutations in this gene, and then they are also delta F508 carriers. So I estimated the probability that any individual would inherit these 5 variants based on the prevalence of Miller syndrome, PCD, and CF in the population. And conservatively, the chance of getting all 5 in one individual would be about 1 in 300 million. 300 billion, sorry, 1 in 300 billion. So almost certainly, Heather and Logan are the only people on the earth that have Miller syndrome and primary ciliary dyskinesia and are CF delta F508 carriers. So that work, the sequencing work, was published a couple of years ago. Another thing that we were able to get from that was the first estimate of the human mutation rate directly from a pedigree. And I've mentioned that that's about 1 in 100 million. So several studies since then have replicated that result. So we now have a good solid estimate of the frequency with which new mutations enter our genomes. That work, we were actually surprised to see how much attention it did get. It was on the front page of the New York Times when the article came out. There's a nice review of the story in Nature published about a year ago, along with a couple of other genome sequencing stories. The Salt Lake Tribune also covered the story. So here's a picture of Debbie, Heather and Logan's mother, and Heather and Logan from about 2.5 years ago. One of the things that came out of that story was the identification of the family. As you know, we're not allowed to reveal the identities of families that we study, but Kirsten Stewart at the Tribune was pretty persistent, and the family identified themselves, and Kirsten realized that they have the same Debbie has the same last name as I do, and that's because she is my wife. So 7 years after I learned about her children in the genetics clinic, I happened to meet Debbie at a party. And it's probably the only time that the phrase postaxial acrofacial dysostosis has been used in party conversation. But it turns out we had a few other things in common as well. So we've been together now for 1/4 of a century. But as you can imagine, for me, and certainly for Debbie and her children, this has been a real exercise in personal genomics. They have now some explanations for their phenotype, something that they have found to be important and useful in their lives. Here's an article that was in Discover magazine a couple of years ago, and this is a rendition in which Logan, who is an artist, is shown with a paintbrush, and then this is a sort of a rendition of human chromosomes. So the idea here is that he could be painting his own chromosomes. Here is some of his real artwork, some of the paintings that he has done. So Logan is an artist, Heather is a writer, and we circulated some poems that she's written you might want to look through. So that's what's on the handout. But I wanted to give you that little bit of background on sequencing of the family, identification of the genes responsible for Miller syndrome in the family. But really, what you all came to hear about is the story, and that's the story that Debbie will tell you. So please join me in welcoming Debbie.